Hey guys, so right now I'm going to be in Raleigh having fun, and you guys are stuck here, so it's awesome. Today we're going to be learning about prokaryotes, and that is what you guys had a little bit of trouble with yesterday, trying to remember what the two domains of life were. And what are they? Anyone? Anyone? They are prokarya and eukarya. The differences being prokarya has a membrane around their organelles, and opposite. Prokarya do not have a membrane around their organelles, eukarya do, and eukarya have nuclei, and prokarya do not. So I'm going to go ahead and move this so I can use the whiteboard, and you guys can see me use the whiteboard just in case if I need to use the whiteboard. Okay, so the first type of prokarya. Let me go ahead and write these down. So we got prokarya. These are all the simple one-celled organisms. That's what we're talking about today. Next type of next domain are eukarya. Those are a little bit more complex, and we'll be talking about them for literally the rest of the class after this week. This week it's only the simple stuff. So we're not going to worry about this, but then we have prokarya. There's two kingdoms inside of prokarya, and the first of which is what we're going to talk about first, and that is archaea. And archaea are extremophiles, meaning they're all of the weird stuff that can live in crazy areas, like in volcanoes or in the thermal vents of the ocean. That's this stuff. They don't breathe oxygen. Sometimes they breathe sulfur. Well, sometimes I'm sure some of them do breathe oxygen, but some of them breathe sulfur. They can go up to really, really high temperatures. They can go up to really, really cold temperatures. They can be in extremely salty environments. So essentially, these guys can live where most organisms cannot. And that's the reason why in evolutionary theory, it's thought that these were the things that evolved first, which is kind of stupid because even though they are more simple in some respects, they have different structures that allow them to bear all of those really harsh conditions. Okay, they also act as decomposers in those extreme environments. They share a lot of characteristics with eukaryotes. In fact, I think they're a little bit more similar to eukaryotes than they are to bacteria. It's just that they still don't have the membrane around their organelles and they don't have nuclei. They share with eukaryotes their methods of replicating RNA and the complexity of their polymerase structures. We're going to move on from that slide. And I've already talked about their extremophile living conditions. So what they have, the structure that they have in order to help them endure all of those extreme environments is a chemical. That chemical is called isoprene. And I think that's going to be on the quiz on Friday. Isoprene is just a really strong chemical. Okay, so just remember that name. They do not have peptidoglycan, and I'll talk about that chemical in a bit. Their transcription and translation of cell processes are different from those in bacteria. And then we get into the categorizations of archaea. The first one is thermophiles, just like the number phile. So phile meaning love, like philosopher is a lover of wisdom, or maybe a francophile is a lover of French culture. Philip means lover of horses. So phile just means lover of. So thermophile means lover of heat, and these are the guys that live in the thermal vents and in volcanoes and all of that good stuff. Then we have methanogens. They are the largest group of archaea, and they are found in anoxic environments like swamps and animal intestines. And they produce barks. which is great. I'm going to try and move this over here so I can look at. Hold on. Never mind, I'm just going to have you guys stay right there. It's just going to be awkward with me going back and forth to my computer, which is currently behind you guys. So anyway, methanogens, they produce farts, so that's really good. They live in animal intestines. Halophiles, they love salt. Halo meaning salt, like halogen, because halogens end up creating salts. They are found in water that is saltier than ocean water, so they live in like the Dead Sea in Israel. Then we have acidophiles, which is self-evident, it means they love acid. They prosper in things like the springs of Yellowstone Park. They were the first archaea discovered. Okay, so we're moving on from archaea, their main things. They were thought to be the first to evolve through abiogenesis, which of course doesn't make sense because of isoprene. Isoprene is the chemical that makes them be as strong as they are. Methanogens are the biggest type of them, or the type of archaea that produces methane, or CH4. And they're extremophiles. They can live in a bunch of different conditions. Okay, so the next type is bacteria. And bacteria is, in some ways, the simplest. Some people say that archaea is the simplest, but bacteria, it shares less with eukaryotes than archaea does. So let me go back through. Okay, so they act as decomposers, just like archaea. They break down old cells, proteins, and lipids. And bacterial structure. They have a bunch of different shapes because it's a really, really big amount of organisms. They have no organelles surrounded by a membrane, like all prokaryotes. And then instead of isoprene, they have peptidoglycan. And peptid sounds like protein. It's kind of a synonym for proteins. And it just makes them really hard to kill. Now, they don't all have it, and they don't all have it in the same amounts. But it does protect the bacteria, and it triggers other cells' immune response system. So that's how we are able to prevent them from killing us. Gram staining is when scientists use a violet dye to classify bacteria into two groups on the basis of peptidoglycan in their cells. So gram-positive bacteria have a greater amount of peptidoglycan and will absorb so much dye that they turn purple. Penicillin affects gram-positive bacteria, so that means that if they die really well, as in if they turn violet really, really well, they're easier to kill. Now, gram-negative bacteria don't have that much peptidoglycan, and ironically, that allows it, that makes it easier for us to kill them using antibiotics. Capsules are kind of like the envelopes of viruses. They are the like mucus-rich surroundings of a bacteria's cell wall, and that protects it from drying out, and that is what allows it to form biofilms. It's slippery, and that makes it them difficult to be engulfed by immune cells. Because they don't have nuclei or a nucleus per cell, they have something called a nucleoid, and that is where the DNA is stored in the bacteria. It's not bound by a membrane again, because none of their organelles are. It's made up of a bunch of circular chromosomes, and it depends how many chromosomes they have is per cell. So it's like no two bacteria have the same amount of chromosomes. And that's due to horizontal gene transfer. Now each one of these chromosomes is called a plasmid, which we learned 2 weeks ago when we were talking about genetics. But again, they're just circular. They're not tightly wound because there's not a whole lot of them. They might have a flagellum, which is essentially a whip-like motor that allows them to spin through a solution. And they also might have cilia, which are hair-like. They're kind of like nose hairs that cover the entirety of the bacteria's cell wall, cell membrane, and then they act like oars to push them through the solution. Reproduction, they pretty much reproduce by meiosis, not by meiosis, but by mitosis. They just divide, which is called asexual reproduction. And when they go through mitosis, it's a process called binary fission. That's another important term for your quiz on Friday. It's the most common form of bacterial reproduction. There's no true form of sexual reproduction in bacteria, but they can exchange genetic information with neighboring bacteria in 3 ways. And the first one is called transformation. That occurs when a bacterium takes in a piece of bacterial DNA that is floating in its environment. So if most bacteria live in a solution, meaning water, so they're just kind of floating in their environment, and then they see this chromosome, for whatever reason, it's a nucleic acid just out in the middle of the solution. Maybe another bacteria died, was lysed, and then just left its nucleic acids out in the ether, kind of like Thor in Avengers Infinity War, where he's just floating around in space, and then the Guardians of the Galaxy pick him up. That's kind of what transformation is. So the bacteria will take that plasmid in, and then take it as its own genome at that point. It will start using those genes. The next type is transduction, and this is a transfer of genetic information from one bacterium to another by way of a bacteriophage. In other words, a virus attacks a bacteria or a bacterium, and just like we talked about yesterday where viruses can add their genes into your genes, same thing. So that would be one possible way that a bacteria could get new genetics. And when this happens, the virus itself is called a bacteriophage. And then the last one is also called horizontal gene transfer, but its formal name for this class is going to be conjugation. And conjugation is where you have two bacteria, they come together, and then they want to change exchange genes. And so they might replicate part of their DNA, and then one of those bacterium is going to create something called a plius. And that plius is essentially a sword because it's sharp and it can poke through the cell membrane of the other bacterium. So the bacteria goes up to it, forms its plius, gets ready to strike, it strikes, and then releases that genetic information into the other bacterium. And now that second bacterium has new genetic information. And that's how bacteria is able to evolve so rapidly. When I talk about evolving, I'm not talking about a bacterium evolving into a multicelled organism that can later evolve into a human. I'm saying that just like COVID is a virus, but the reason why COVID, influenza, there's another one that I'm forgetting. Anyway, the reason why those specific viruses, are so hard to control is because they can change genes so rapidly, and they can have such a wide variety of different